basically two different geometrical solutions to the same problem. And there's no hidden significance or anything like that. It's just that one, they come in from opposite directions basically, and that's what results in getting the 2' OH in one case and 3' OH in the other. But the editing mechanism exactly the same, except the CCA end of the RNA flips opposite directions to get from the editing site to the catalytic site. And so that's all it is, just interesting geometrical differences.
In the formation of the peptide, what happens if the mRNA and the mRNA never reach a stop codon? For example, if there's a mutation or like poly A tail is added before the stop codon, then what happens to the protein?
I didn't quite get, what happens if, what kind of mutation?
When we are making peptide and the translation, well, we are reading the mRNA, but what happens like if there's no stop codon on the mRNA that's being read by the ribosome?
You mean if there's a mutation that converts the stop codon to something else that codes for an amino acid?
Yes.
Yeah, that's actually an important evolutionary event because, well, first of all, it can be very damaging to the organism, but it may not be. And that's because all that happens is the ribosome continues on translating. So it makes the original protein, and then it adds on a whole bunch of new amino acids. Sometimes those new amino acids fold up into their own separate domain and don't affect function. It just makes the protein a little bit more, a lot more energetic to make because now it has to go until it finds another stop codon downstream. Other times the new amino acids can completely mess up folding of the protein and it can be a lethal mutation. So it just depends on the nature of the new amino acids that get added when the ribosome continues. But that's, if you take a stop codon and convert it into something that codes for something, it's a mutation for sure, and it can have major consequences. That's the converse of if you mutate codon that codes for an amino acid, converting it to a stop codon. Same thing in reverse, or converse, where the ribosome will stop before the protein is finished.
So we don't have any mechanisms that can recognize that in the protein and kind of chop off the part that is extra.
You're asking protein synthesis to have as rigorous error correction as replication and transcription, basically, right? You want something comparable to mismatch repair and all that sort of thing to happen to proteins as well. There are multiple answers to that. Maybe the most important is that for protein synthesis, the occurrence of errors like that is usually let me say it differently. Well, yeah, the occurrence of errors like that might kill you, but it won't get passed on to your kids. And so evolutionarily, it's not such an important thing that it absolutely must be corrected. Errors in DNA get passed on to progeny. And so if there's an error in translation that arises during translation, that's not such a serious thing. There are many polypeptides being made. If it's a mutation that wasn't caught so that there are two reasons. I just said the first reason, which is if it's an error that occurs during translation because of a mistake during translation, that only affects that polypeptide. It only affects you, it doesn't affect your progeny. And so it's, evolutionarily speaking, it's not a big deal. The other thing is that if it arises from a stable mutation that gives rise to this error, And if that mutation was not caught by the DNA polymerase, and was not caught during the original coding, was not caught during editing, was not caught by mismatch repair, and was not caught by subsequent repair, I mean, there you've taken just about everything imaginable to try and make it accurate. And if the mutation got through all of those barriers, by the time you get to translation, you're dealing with the conceptually most complicated system in which to try and catch errors. Because once the protein is made, instead of 4 bases, there are more than 20 amino acids. And so that's a much more complicated error correction thing. And proteins are designed to fold in thousands of different ways. And so it's hard to imagine an error correction mechanism that could catch a mutational error in one particular protein. There is protein error correction, but it's for more generic types of errors. For example, if proteins get damaged in terms of their configurations around their peptide bonds, if L gets converted to D or so on, there are enzymes designed to recognize that. There are mechanisms for taking proteins that are not folded properly and getting rid of them. And so if any of these errors give rise to proteins that are not folded properly, they can be got rid of. They won't be corrected, but they can be got rid of if they're not folded properly and so on. But it's so complicated at that level, you couldn't correct a single error. And so if it hasn't got it at the DNA level and the RNA level and all these repair levels, then forget it. Might as well let you die and let somebody else carry on.
Going back to slide 123, where we talked about the evidence for the triplet code and the non-overlapping code. I'm a little confused about the rationale that they use and like how the process occurred with the proflavin.
You're confused about why proflavin indicated that it's a non-overlapping rather than an overlapping group. Okay, so think about what is the most serious problem that you could get from a proflavin mutation if it's non-overlapping and if it's overlapping? If it's non-overlapping, which is basically just this line here, if you take this C and get rid of it, then this codon is shifted up 1 notch to this position, and the first codon becomes, instead of AUAC, it becomes UAU, and every codon afterwards is changed. And so this is a frame shift mutation, which is the most disastrous kind of mutation that is possible, because the entire protein is messed up. Every single amino acid is messed up. If it were an overlapping code, and you got rid of this C, codon 1 would get messed up. Codon 2 would be this one, and that would be messed up because the C is missing. Codon 3 also contains the C position, so it would get messed up as well. Wait, no, codon 3 would be okay because UAC would be down in this position. And then codon 4 would be ACU, which would be codon 5 here before the frame shift. And so all that happens is all the subsequent codons are shifted 1 position up, but they're all still there. So the only amino acids that get messed up are the first 2 in the protein in this particular example.
Didn't 3 mess up as well because originally it's AC or CUA? No?
It's CUA here, and yes, that, I'm sorry, But starting with codon 4, the only difference is that it is up here in the third position instead of CUA. And so basically, by eliminating this base, you have messed up 2 codons and lost a codon, and everything else is okay. Now, of course, it might be that the first 2 codons are for very important amino acids that are critical to protein function, and then of course you lose the function, but that's not very likely. I mean, it's not, if you look at a protein with yay many amino acids, the chance that any one of them is going to be absolutely critical to function is small.
So then, could you remind me again what the proflavin is? It gets inserted into the DNA, is that right?
Yeah, you can look at the structure, and from a sort of a broad global perspective, it looks like a base. It's planar, it's electron rich. Obviously it's going to stack very well with itself or with bases. And it obviously is very hydrophobic and it's going to avoid being floating around in aqueous solvent. And so the moment it encounters a nucleic acid, it just wiggles in between the bases and goes to sleep.
Okay, so once it gets inserted during replication, the mutation arises because of a random insertion of a base. Is that what you said?
So once the proflavin is sitting between the bases and that comes up to the polymerase, if that's in the coding strand, or in the template strand, I should say, if that's in the template strand, when the non-template, when the growing strand comes to add a base where the proflavin is sitting, the polymerase doesn't know what to do. And most of the time, it'll be confused and sit there, and maybe the proflavin will diffuse away and the problem might go away. But there's always a chance that a base will get inserted at the position of the proflavin, and it'll just be obviously any random selection of the 4 bases. And so your chance of having a mutation is 75% wherever that proflavin is sitting. And if it's reversed, if the proflavin happens to be intercalated in the comes in and intercalates itself in the growing strand, it can have the opposite effect.
Can you explain how do they determine that it's arrived by every 3 codon or more?
That's summarized in this part of the figure, where they found that when they made a lot of mutations with proflavin, these are in bacteriophage, so they got hundreds of mutations. It's very easy to work with. You can do this whole experiment in a day. They were able to generate hundreds of mutations. They were looking at just one particular protein. They generated hundreds of mutations in this one protein. And then they did crossover experiments. They took 2 different mutant phages and allowed them to infect E. coli at the same time. And their DNAs would cross over during replication. And some of these crossover events would yield 2 mutations in the same DNA, because the crossing over takes place between the 2 mutations. And so what they were able to find out was that they could group all of their mutations into 2 groups. When they did this crossover event, they found that about half the time, the crossover events, well, not less than half, but a significant number of times, when they did crossover between two different mutants, it would generate a protein that was functional. In other words, the two mutations rescue each other. If there's a crossover event between them, that puts them on the same DNA strand. So if they took and classed all the mutations according to those that could rescue each other and those that could not rescue each other, they got two classes. Just two. One, all the mutations in class A, each of those mutations could rescue any of the mutations in class B. And so they explained this, they rationalized this by saying they had a pretty good idea how proflavin worked, and this validated that idea, that proflavin causes deletions or insertions. Oh, and that's about the proflavin mechanism. Of course, one thing I didn't emphasize was that if you have the proflavin in the template strand and it puts in a random base, that is going to be an insertion mutation in the final analysis because a base has been put in where the proflavin was, and then it moves on to the next real base. And so proflavin gives either insertions or deletions. And so they had a hunch to begin with that if they had one mutation that was an insertion, and if they paired that with another mutation that was a deletion, the two would cancel each other, as suggested here. So if you delete this C, then this whole thing goes up so that, for example, codon 4 here is messed up. Should be UAC. But if you add another mutation in, that's an insertion. So you delete a C here, and if you add an N here, you regenerate the original frame. And so everything downstream then is correct. So they had these two classes of mutation. Each member of one class could rescue each member of the other class, meaning that one was insertion, one was deletion. They didn't know which was which, but they knew that one was insertion and one was deletion. Then they found that all of the mutations in one class could rescue themselves if there were three. If there were three, they could rescue themselves in either class. So it took just one to compensate between classes, but it took three within a class. So they said the rationale for that is essentially the same. If you take three additions, and if you take three addition mutations, insertion mutations, and put them together, and you rescue protein function, it must be that each mutation is the insertion of a single base, which gives you a frame shape. But when you put three of them together, all you do is create an extra codon, but you restore the reading frame. And same thing with deletion. So it was all based on rationale emanating from the presumed mechanism of action of proflavin in causing either insertion or deletion mutations in the DNA. Is that? Yeah.
For the addition, how likely will they? You said you create new codon, right? How likely will that codon be together instead of all over the places, different three places?
Yeah, you're saying what happens if there's a mutation here and one way down there? They would compensate ultimately, but if the final compensatory mutation doesn't occur until near the end of the protein, what good is it going to do? That's what you're saying, right? And that's true. So that's why I backtracked earlier when I said that half of them would compensate. It's not half, it's less than half, because sometimes they're too far apart and so it doesn't do any good. So the compensation has to be early enough in the protein structure.
For the three at the same time, check within the same class.
three mutations onto the same DNA molecule.
On yesterday's lecture, you were talking about how to identify whether the polypeptide is synthesized from N-terminus to C-terminus. Can you explain the experiment where you use carboxyl, you digest it with carboxyl peptidase? It was on the slide 134. Yeah, you say you digested with carboxyl peptidase?
Yeah. So remember, this experiment was done in 1961. Where are we? It's a famous experiment, very famous. It was a huge advance at the time. But this was a long time ago. And so the techniques were extremely primitive compared to what's available nowadays. And these numbers of the peptides refer to numbers based on the techniques they use to look at them. So here, after digesting the protein and running it on this two-dimensional thin layer chromatography, paper chromatography actually, you notice that they just numbered the peptides 4, 5, 6, 7, 8, 9, and so on. These numbers having meaning only with respect to the method used to separate them and having no significance at all with respect to their order in the protein. So what they said was that if our experimental rationale here is correct, then we ought to be able to get an answer for each one of these peptides, which will rank the peptides monotonically in something that can be graphed like this. From the peptides at one end of the protein should tend to have very little label. Peptides at the other end, a lot of label, and there should be a gradation, a gradual increase all the way through. So we ought to be able to take these peptides and rank them. And that's what they did here. There aren't a whole bunch of low ones and a whole bunch of high ones, for example. Or they're not a whole bunch in the middle, but rather they could rank them in a nice straight line here. And that was very exciting to them. That alone was very exciting to them, that they could rank these peptides like this. They didn't know the sequences of these peptides at the time. They just knew that they were individual peptides in the protein. But this, in fact, can be used to sequence the peptides now because this tells them what the order is. The only thing is they didn't know whether this was the N-terminus or the C-terminus, and that was the entire question that they wanted to ask. Does synthesis go from N-terminus to C-terminus or C-terminus to N-terminus? So they've arranged them in order here, but they didn't know whether this or this was the N-terminus. That's why they used the carboxypeptidase. Because carboxypeptidase digests proteins beginning only at the C-terminus. And if they treated the protein very briefly with carboxypeptidase before conducting the peptide mapping, then the peptides at the very end should be gone. For example, if this peptide has 7 amino acids in it, and you cut off just one of those 7 amino acids, it's going to migrate differently on here. And so the peptide will be lost. For example, number 16 here, you just barely see it, but it's a light peptide. Light peptide. But after carboxypeptidase treatment, these other peptides were unaffected, but 16 was completely gone. So that meant that 16 was at the carboxyl terminus. And so then they were able to say, synthesis proceeds from N to C.
Was it gone because the carboxypeptidase digested all the label using?
Well, carboxypeptidase removes amino acids one at a time from the C terminus of a protein. That means that any peptide that occurs at the very end of the protein will immediately be degraded and lost if treated with carboxypeptidase. And so they just have to look, if we treat very briefly with carboxypeptidase, which is the peptide that's lost first? The one that's lost first must be at the C terminus.
Can you quickly go over the experiment, the initial experiment, only the first part?
So the experiment started with grinding up, well, not grinding up, they probably started with red blood cells. But they started by lysing the cells. They started by lysing the cells and collecting the homogenate that resulted from breaking open the cells. And then they removed the ribosomes by centrifugation, probably. Or else they might have done it on a sucrose gradient. It doesn't matter what the technique is. They isolated the ribosomes. There are various ways of doing it. And washed them so that the only thing they had was the ribosome and anything attached to the ribosome. And of course, attached to the ribosome will be partially completed polypeptides, because the ribosomes were collected, the different ribosomes represented all stages of polypeptide growth for this one protein, hemoglobin. Then they took those washed ribosomes with partially completed polypeptides on them and resuspended it in a mixture, cytoplasmic extract, basically, that contained all of the necessary enzymes for charging amino acids and for carrying out protein synthesis. And then they put all of the amino acids, they made sure that this mixture had amino acids in it, including labeled leucine, so that they could so that they could identify the peptides based on the radioactivity that they contain. Could have been any amino acid. Leucine was chosen for practical reasons. And then they allowed a short time of synthesis to take place. The idea being that any proteins whose synthesis was completed during this short time would be released from the ribosome. And then after this short time of synthesis, they collected the ribosomes again, threw them away, and looked only at the proteins that had been completed on the ribosome during the course of the experiment. And then they said, proteins that were completed during the course of the experiment will range from those that were almost finished to begin with and just had a couple of amino acids added, to those that had been started only a short time earlier and had a lot of amino acids added. And then they did the digestion with trypsin and the paper chromatography to separate the peptides, and looked at how much radioactivity was in each of the peptides in order to eventually come up with this graph.
Can you go over how for your cell blocks the primary synthase mechanism?
Okay, your question was about
How for your cell blocks
how fluorouracil messes up this mechanism. Yeah. So the basic strategy. If fluorouracil can be said to have a strategy. The basic strategy by which fluorouracil inhibits the thymidylate synthase is to pretend it's a substrate, invite the enzyme to accept it into its active site, and kill the enzyme. Or actually, because the enzyme attaches fluorouracil in its active site and then can't do anything, this is often called a suicide inhibitor because it causes the enzyme to commit suicide. And the way the enzyme commits suicide is because fluorouracil gets attached here in exactly the same way as the uracil does. And it can attack the tetrahydrofolate in exactly the same way the fluorouracil can here. It can attack in exactly the same way as the uracil to give us this tetrahedral intermediate. Problem is that fluorouracil sitting here now has a fluorine. And a fluorine does not want to be abstracted as an F+. You know that they're very electronegative. So a fluorine is absolutely not going to come off as an F+. And so it sits here. Can't go anywhere. The enzyme has committed suicide by attaching fluorouracil in its active site and getting stuck. The enzyme has committed suicide, and this one fluorouracil has inactivated the entire enzyme for good.
What happened to the bad enzyme?
What happened to the what?
What happened to that enzyme?
I'm
That
sorry?
enzyme? What happened to it? Did it just,
What happens to the enzyme?
like, did it
Oh, it's just turned over like all bad proteins in your system. You've probably heard the old saying that your body turns over once every 7 years or something. It's not very accurate, but what that means is that there's constant protein turnover in your body because of proteins that get damaged or whatever it might be. And so this protein, the inactivated enzyme, will eventually get degraded. And the amino acids will be reused. And the fluorouracil, at some point the fluorouracil will eventually get degraded by one of the pathways that get rid of free bases. But it might live to inhibit another enzyme someday, but at some point it's going to get completely degraded. So everything gets degraded eventually. Of course, if this is being administered as part of chemotherapy, and this enzyme is in a cancer cell, the whole cancer cell dies. And so it's not really an issue what happens to the enzyme because the whole cell is dead. Because it's impossible to make any thymine, and without thymine the cell can't grow, so it just dies.
Is competitive inhibitor is always a suicide inhibitor, or it's the other way around?
A suicide inhibitor and a competitive inhibitor are really two completely different things. You might consider a suicide inhibitor as competing for uracil. For example, fluorouracil competes for uracil to get into the active site. But because it competes for the uracil, does not mean it's a competitive inhibitor, because competitive inhibitor is a very technical term that refers to the mechanism of inhibition. A competitive inhibitor, by definition, is an inhibitor that inhibits only by virtue of its ability to compete out the substrate. The enzyme is never inactivated or damaged in any way. When you remove the competitive inhibitor, the enzyme is still fine. So the fluorouracil does compete with the proper substrate to get into the active site, but it's not a competitive inhibitor. So a suicide substrate is not a competitive inhibitor. Is a competitive inhibitor a suicide substrate? No, it can't be, because a competitive inhibitor never inactivates the enzyme. Does that answer your question?
This has probably been asked before, but are we supposed to like be memorizing these mechanisms and stuff?
No, you shouldn't memorize any of the mechanisms. You need to understand them.
And the pathways?
And the pathways, yeah. So be able to walk yourself through how they work. And some of them guided by how much time we spend in class and the importance attached to them in the context of the course and so forth. Some of them you want to pay more attention to than others, but you want to understand them completely, because the reason I say don't memorize them, there are two reasons. Number one, I will never ask you to reproduce the thymidylate synthase mechanism. I'll never ask a question like that, because it's a moron question. You can memorize it without understanding a thing. The second reason is that if you're trying to understand the mechanism, and one way of trying to understand the mechanism is in fact to draw it out on a blank piece of paper. Do I really understand how this pathway works? Let me see. Because if I understand it, I'll be able to reason my way from one step to the next. So you try and draw out the whole mechanism, the whole pathway, whatever it is. If you've memorized it, you'll never know whether you understand it or not, because you remember what the next steps are. And that won't help you at all if I give you an exam question that says, here's a mechanism that shares some similarities with what we studied in class. Figure out how it works. If you've memorized them in a class but don't understand it, you can't answer the question.
Would memorizing it help, or is it going to be useful?
If you need to remember something, I'll give it to you. Memorization will always hurt. Really. It'll always hurt your understanding. They're in conflict with each other, quite literally. If you remember something, you have no way of knowing what you understand unless you find new mechanisms to look at and try and reason your way through it. Memorization is sometimes important for being able to do things quickly. Like if you need to reason through something, but in order to do that you need to have a lot of facts at your disposal, then it's useful to have it memorized. But when I recognize a situation like that, I give you the facts in the exam. So I try to avoid anyone ever being inhibited because they didn't remember enough facts.
Professor, on slide 116, there's a picture of reductase. Oh, right, can you briefly go over how you mentioned the radical stabilize with the tyrosine and the cytosine? One more. Oh, not the mechanism. Like this one? This one. This side.
What is your question?
Can you explain how you mentioned the radical is stabilized within the riboglucoside reference? You talked about tyrosine 730 is allowing for the radical formation of the cytosine.
Okay, so this is the radical generating center, starts with iron. And so it stabilizes quantum mechanical mechanisms that we're not going to discuss here at all. It stabilizes the free radical here on this tyrosine. And that tyrosine is this tyrosine 122.
Oh,
And this tyrosine 122 lies near tryptophan 48, which lies near tyrosine 356, etc. It's just like electron transport, really. And so ultimately, this iron center down here, through this chain, is stabilizing this cysteine 439 in its radicalized form. And that's what you see here. This is probably that cysteine. They're not absolutely sure yet, but most people don't worry about it because the evidence is so strong.
Back to the secret cases, you said the difference was that the class two flipped over to the acid side. So does class one just have that, you just showed the reaction, so it didn't flip at all? Or just
I don't understand.
So the image for the class two shows the two different, slide 131. Yeah, the two arrows, does that indicate the flipping?
Yeah.
And then, for the one with class one, it was for the image for class one in the previous slide, in the 3D image, for the editing side, does the class one just not have the editing event into the editing side, or does it?
Yeah, they both flip. They both flip. It's just that in one case, this is the editing site and that's the active site. In the other case, that's the editing site and that's the active site. Just because the geometry is reversed, but it has no significance.
Do you hold a review session or a final?
Yeah, I'll have one on Sunday again.
Last question. Can you explain the equation on the previous slide?
The equation itself is not really what concerns us here. So I'm not gonna go back to 153A level thermodynamics. But I put this here just to emphasize that there is a rigorous thermodynamic limit to how accurately an enzyme can choose between isoleucine and valine. And it starts with the equilibrium constant up here. The free energy change for any reaction related to its equilibrium constant that you learned in 153A through this equation. And if you rearrange the equation and take a ratio for reaction 1 and reaction 2, you'll get a ratio of the energies, basically, for binding this versus binding this. And that's related to the ratio of the equilibrium constants. If it binds more tightly, the equilibrium constant will shift towards the bound mode, right? And so you can see easily that if you take a ratio of the equilibrium constants between two reactions, you'll get an estimate of the amount of energy, not an estimate, you'll get the energy difference between those reactions by using these equations. So that's the rationale for it. And what this says is that the possible level of discrimination, the best level of discrimination possible thermodynamically for the enzyme to choose between isoleucine and valine is the ratio determined by the binding energy possible for one methyl group. It's absolutely impossible to discriminate any better than the amount based on the binding energy of one methyl group, and that turns out to be about 100-fold. So the enzyme can distinguish between isoleucine and valine to the extent of 100-fold and no better, just based on binding. So most of the discrimination comes from the editing step. It's a very expensive way to get accuracy, but it's the only way